Hello students, I'm Dr. Arpita De. A very warm welcome to all of you. The topic for today's discussion is ultrastructure of Golgi complex. Golgi complex is otherwise known as Golgi bodies, Golgi apparatus, dictyosomes, you can call it by any term. Golgi complex is a compact, discontinuous system of membranes that are found to occur in the cytoplasm of the cell. This organelle is an indispensable one in protein trafficking mechanism of the cell. This organelle can also be called as the traffic controller, traffic police of the cell, because it categorizes and channelizes proper proteins to its respective destinations. It was discovered by Camillo Golgi in 1898 in animal cells. It is also known as dictyosome in plants. This is an electron microscopic picture which shows the compact nature of Golgi complex along with its vesicles and cisternae. Golgi complex is made up of four components: the cisternae, Golgian vacuoles, tubules, and vesicles. We will learn about each one of these in details. The most prevalent theory of how Golgi apparatus forms is the cisternal maturation model, which suggests that the sacs themselves tend to move from the cis phase to the trans phase of Golgi apparatus over time. Now let's talk about the components that make up the Golgi apparatus. Let's start with vacuoles. The vacuoles are modified and expanded flattened sacs occurring at the edges of the complex. So they are normally found at the peripheral part of the complex. Now coming to tubules. These tubules are anastomosing in nature and they have a diameter of 30 to 50 nanometer. They surround the periphery of the dictyosome and radiate from it. Please note it is dictyosome. Please check this spelling error. Then coming to cisternae. Cisternae are about 1 micrometer in diameter. These are unit membrane, smooth, that is without ribosome. They are unit membrane bound, central, flattened, plate-like or saucer-like closed compartments with dilated edges. Please remember, with dilated edges. They are held in parallel bundles, one above the other. Cisternal spaces are also there in between. You can see their arrangement from this diagram. The cisternae are arranged in parallel bundles, one above the other, and in between cisternal spaces are there. Dictyosome has polarity. The convex end of the dictyosome comprise proximal forming or cis phase, and the cisternae at the concave end of the dictyosome comprise the distal maturing or trans phase. So this is the concave side, which is the trans phase, and this is the convex side or the cis phase. Now let's come to the vesicles. The vesicles are of three types: transition vesicles, secretory vesicles, and clathrin coated vesicles. The vesicles normally have a diameter of 60 nanometer. Transitional vessels are small membrane limited vesicles which are thought to form as blebs from the transitional endoplasmic reticulum. They migrate to and converge with the cis phase of Golgi, where they coalesce to form new cisternae. Cis phase means they will converge with the concave side of Golgi apparatus and they will join with each other. That means they will coalesce with each other to form new cisternae. Then comes secretory vesicle. This is very important. The secretory vesicles are what? The flattened sacs bud off or pinch off to give rise to this vesicle. So these vesicles rise by virtue of pinching off or budding off of the flattened sacs. The smaller membrane-bound vesicles are aggregated around the stacks of cisternae. The vesicles fuse together to form secretion granules which fuse with the plasma membrane to discharge their contents by exocytosis. So these vesicles will form, will bud off from here. They will carry the required cargo in them and they will do the shipping activity. Shipping means what? They will pick up the cargo from the source and they will take it to the destination. They will travel to the plasma membrane and then they will fuse with the membrane and discharge their contents by the method of exocytosis. Finally, coming to clathrin coated vesicles. These are spherical protuberances about 50 micrometer in diameter and with a rough surface. They are found at the periphery of the organelle, usually at the ends of single tubules. The clathrin-coated vesicles are involved in intracellular trafficking of membranes and of secretory proteins between ER and Golgi and vice versa. That means what? Anterograde and retrograde transport. Also between the GELR region and the endosomal lysosomal compartments. Thus, Golgi complex is the traffic control of the cell. This anterograde and retrograde transport is not being discussed in detail over here since this video is made to study the structural details of Golgi apparatus. So we are not emphasizing on the functional details in this particular lecture. So this is all about the structural details of Golgi complex. These are the few references from where I have downloaded some of the pictures. So this is how we end our lecture here. If you have any doubt or confusion, please feel free to get back to me. Share this video with all your friends, those who are in need of it, and please make your own notes from it, because always remember, the more you write, the better it is, and you will learn better from writing. So hope to see you soon in our next upcoming lecture. Thank you so much for your kind attention.